So let's move on to meiosis 2. And meiosis 2 is going to be the phase that's most similar to mitosis. So we're going to go ahead and we're going to start here with prophase 2. And I'm showing 3 dyads or 3 chromosomes which resulted from one of the cells at the end of meiosis 1. Now, I want to make a point to tell you that even though I show this chromosome as being all red and this one all blue and this one all blue, they're really kind of plaid at this point because crossing over has occurred. For simplicity, I'm going to draw this one all red. Just know that this one has a lot of red in it with some blue that resulted from crossing over. And the same thing goes for these two over here. So in prophase 2, the nuclear envelope begins to break down, the spindle apparatus begins to develop or form, and the chromosomes begin to condense. This is after a brief period of interphase, or sometimes no interphase whatsoever. Sometimes we just go from meiosis 1 straight into meiosis 2. And sometimes there's a very brief interphase. Now, if there is a very brief interphase, no S phase occurs. So the chromosomes do not replicate again. We skip that. We just go from meiosis 1 pretty much right into meiosis 2 without any replication of chromosomes occurring. Now we're going to go into metaphase 2, and this is where the dyads line up on the equatorial plate. Again, there's no independent assortment here. That only happens during meiosis 1. So the chromosomes line up on metaphase plate or equatorial plate, and they get ready to move to opposite poles. In anaphase 2, we show just that. The centromere actually breaks down here, and the chromosomes begin to move towards opposite poles. Then we make it to telophase 2, and this is where the chromosomes have moved opposite poles already. The nuclear envelope begins to reform. The chromosomes begin to decondense or relax, and the nuclear envelope reforms, spindle apparatus disappears. And again, at the same time, a separate mechanism, cytokinesis, is occurring, and the cell is literally dividing right in two. And that's it for meiosis 2.